In this video tutorial, we will be discussing about what actually is gene structure and why we perform gene structure analysis. So basically, there are several questions which might come in one's mind when dealing with the gene structure. For example, when dealing with the research related to gene families in crop plants, questions like why we do gene structure or intron exon structure analysis. What if there are more number of introns in a gene? Now let's have a look on these type of questions one by one. Firstly, we look on why we do gene structure or intron exon structure analysis. Basically, we do this type of analysis to deeply understand the phylogenetic relationships, genetic variation, and potential protein functions among the member of a given gene family. There might be some other type of questions that what if there are more number of introns in a gene family or in a gene. Basically, introns have major role to play in the evolution of various plant genes, and many gene families indicate less or lack of introns in them. So these intron exon structural differences might be due to insertion deletion events and are helpful in the identification of evolutionary mechanism. And in general, introns are considered to be under weak selection pressure, and the absence of introns indicate that genes might have evolved relatively rapidly. Furthermore, the number of introns present early in a gene family evolution were reduced over time, resulting in gene families with fewer or no introns. And those gene families which have less number of introns are considered to be evolutionarily advanced, while gene families with more or a larger number of introns are thought to have gained new functions during evolution. So this is all about gene structure and some basic questions which might come in one's mind when dealing with the gene structure analysis.